This is the MSH2 lab abstract.
My name is Elsie Macharia.
My name is Gaina Yvonne Pierre.
My name is Unique Hayes. And our professor is Dr. Chen. This is Biology 125, Section 4, Group 2.
During the duration of our lab, there has been a series of procedures involved to determine whether the mutations in MMR or mismatch repair genes increase susceptibility to HNPCC, which is hereditary non-polyposis colorectal cancer. The mismatch repair genes are genes MSH2 and MSH6. We are going to focus on MSH2 in this lab. The lab was conducted through a series of procedures including plasmid isolation, digestion, and gel electrophoresis. The objective of our lab was to use molecular techniques to investigate how mutations in the MSH2 gene affect mutation, as well as identify mutations within a target DNA sequence in order to investigate the connection between MSH2 and HNPCC. This inherited disorder increases the risk of colon cancers. What's the connection? Mutations in certain genes, especially the MSH2 gene, has been said to increase the risk of developing HNPCC.
The following materials were used when performing the labs. For plasmid mini prep, we used 5 ml of E. coli overnight culture, which were 3 different strains, and Zippy plasmid mini prep kit. For restriction digestion, we used double distilled water, the 3 plasmid DNA samples prepared from mini prep, 10 times fast digest buffer, and the enzymes XPA1 and APA1, and 1% agarose gel. For PCR, we used the 3 plasmid samples from the mini prep as well, a solution containing 10 micromolar of both forward and reverse primers, 3 PureTac ready-to-go beads in 0.2 ml PCR tubes, and nuclease-free water. For restriction digestion of PCR product, we used DDH2O, the 3 PCR reactions prepared during PCR, the restriction enzyme BSMA1, 6 times loading dye, 1.5% agarose gel, and 10 times RE buffer solution. The experimental design was as follows. For plasmid mini prep, 3 different strains of E. coli were cultured and used to produce more plasmid DNA that will be used for downstream experiments. The Zippy plasmid mini prep kit will then be used to isolate plasmid DNAs from the 3 E. coli strains. Restriction digestion. This will be used to tell the difference between the empty vector and the 2 plasmids containing an MSH2 insert, 1 wild type and 1 mutant. After digestion and analysis of the agarose gel, the plasmids with the insert will have similar patterns, 2 bands, while the empty vector will typically have just 1 large band. PCR. This will be performed to amplify a region of the MSH2 gene. Since the empty vector does not have an MSH2 insert, there should be no fragments produced by PCR. Restriction digestion of product. This will be done to enable distinction between wild-type and mutant MSH2. Analysis of the agarose gel should reveal no bands for the empty vector. The wild-type MSH2 will be distinguished from the mutant because restriction enzymes digest them differently. A determined enzyme, BSMA1 in our case, will digest the mutant but not the wild type. The gel should therefore show more bands in the mutant than the wild type.
The methods. Overview. This entire lab is to determine if MSH2 is linked to HNPCC. We have determined the identity of the plasmids as well as our mutant. SPO is the blank, which is PRS413. SP1 is the wild type, which is MSH2, and SP3 is our mutant, which is AG131. We isolated the plasmids by using the Zippy plasmid mini prep kit, and then we used the nanodrop method to determine the absorbance and concentration of our plasmids. We used gel electrophoresis to determine which plasmid contained the MSH2 insert after we digested the plasmids with the enzymes APA1 and XBA1. Then we used PCR to determine the region of the MSH2 insert. Then we use the restriction enzyme BSMA1 to determine which plasmid contained the mutant MSH2 by using gel electrophoresis. These are our plasmids. We had SPO1, SPO blank, and SP3. The concentration for SPO was 160.8, which is a decent amount of concentration. The concentration for SP1 was 63.4, which is a concentration that had too much alcohol in the PCR digestion. SP3 had 284, which is also a good concentration. And the purity for the strands were all 1.8, 1.7. So that goes to show that the purity levels of the concentration were very well together. And as a result, when we did the gel electrophoresis, we had to label them. And we broke the original strands, the original 3 strands, into cut and uncut. The cut strand had enzymes, whereas the uncut did not. And we used it to do gel electrophoresis.
And so the image you see on the right shows how we determined which enzyme we will use to tell the difference between our wild type and mutant. As you can see, in the wild type, we don't see BSMA1, but we do see it in the mutant. Please note that FAT1 and BCOD1 could have been chosen as well, but because they are more expensive, we chose to use BSMA1.
Here is a picture of our gel electrophoresis. As you can see, this here is the KB ladder. This is the SPO uncut, and this is the SPO cut. As you can see right here, that is where we have the MSH2 insert. Right here is SP1 uncut, and it was disappeared because we accidentally punctured the well. There was a possible error within this lab. And here's the uncut, cut, uncut. So to determine the size of the PCR product after the digestion of the restriction enzymes, we were able to determine that our restriction enzyme was BSMA1, and we separated our plasma sequences by their identifying name. As you can see, SPO was the blank, SP1 was the wild type, SP3 was the mutant. Once we put this into APE, the size of our PCR product was 883. After we digested the plasma with BSMA1, for the wild type we had no bands, but for the mutant we had 1 band.
And this right here shows how we digested using APE. For MSH2, again, there should be no cutting sites because BSMA1 does not cut MSH2, so you would just get 1 band. But then for the mutant SP3, BSMA1 does cut it once, so you would have 2 fragments, 1 large one and 1 small one. And so when you look at this picture, the first 2 lanes are the empty vector. Since they don't have an MSH2 insert, there would be no fragments to observe. The next 2 lanes were the wild type, uncut and cut, and then these 2 were the mutant, uncut and cut. It's not very visible, but you clearly see there's a difference between the wild type and the mutant, and that's because BSMA1 cut the mutant and not the wild type.
So during this lab, we investigated how mutations in the MSH2 gene affect the mutation. We diluted and digested DNA to identify MSH2 insert in our samples through electrophoresis, and at the end of the lab, we found that our MSH2 was within our 3rd sample. Some possible errors that did occur was one of the wells was punctured, thus not allowing the gel to travel. It did not contaminate our results because it was an uncut sample, therefore not affecting our end result. Possible errors that did not occur but could have was incorrect pipetting or loss of the agarose gel when pouring gel into tray. Incorrect pipetting was not a problem for our lab because it was shown at the end that all of our devices were measured correctly. We learned at the end of this lab to use restrictions enzymes in order to identify a mutation within a target DNA sequence, increase our pipetting skills, increase the ability to make agarose gels and run gels, the use of RCETA for scientific and mathematical calculations, and the use of APE in determining the mutants by using the translation function.
Future directions. Next week, we are going to perform a yeast transformation using AGY75 without mutant MSH2. The week afterwards, the yeast transformation lab, we will use the FOA assay to measure the mutation rate in the yeast that contain the plasmids. We would like to acknowledge the Spelman College Biology Department. We would like to acknowledge Dr. Hong Chen and Dr. Stephen M. Kyoko for patiently working with us during this lab. We would like to acknowledge Guyana Yvonne Pierre for her hard work and dedication in organizing the slides as well as providing pertinent information for the slides detailing the introduction and discussion. We would like to acknowledge Elsie Macharia for joining our group and being a dedicated member since joining, also for including the experimental design as well as the understanding of the graphs and data. We would like to acknowledge Unique Hayes for her hard work and persistent attitude. We would like to acknowledge Unique for her role in grammatically correcting the information as well as the insertion of the data and slides. And here are our references. We use an article that was found on PubMed with the ID 194669295 and the Spelman College Department of Biology Moodle's comment page. Thank you.
Thank
Thank you.
you.